Okay, I think it's time to start, and I'm very glad and happy to welcome all of you. My name is Sandro Zampieri. I'm the director of the Department of Information Engineering that is promoting this series of distinguished lectures. This is, let me introduce a little bit why we propose every year this kind of lectures. These lectures should be of wide interest and should be also some opportunity to see what we consider very hot topics, both in our field and in the fields that are on the border of our discipline. And so in the fields in which we see some opportunities of interactions. So this year, this is the first lecture we are proposing. The next two, one will be in this month, at the end of May, and the other will be in September. This lecture will be on the interaction between engineering and health. The next one will be also very, very wide and about wireless services and smart cities and health again. And the last one in September will be on learning and machine learning and deep learning, another very hot topic now. And so let's say we hope that you will enjoy this opportunity and this lecture. And today we have, I'm particularly happy to welcome one old friend, Paolo Vicini. I'm a little bit older than you, but we were PhD students together many years ago, we have to say, even though we could, when we have seen yesterday, we could recognize each other, and so we were very happy that at least we are able to see something similar of the face we had 20 or 30 years ago. And so this is at least something that we can be happy about. I'm very happy to have Paolo again here. He had his trajectory was completely different. You know, he went away very soon from Padua and went to other institutions, but his trajectory was very successful, and so I'm very proud to have him back here and to have the opportunity to listen to him. Thank you.
So good morning. I'm Giovanni Sparacino. I'm a professor of biomedical engineering in this department, and it is truly a pleasure to introduce today's speaker, Paolo Vicini, and his lecture. The title is both brilliant and self-explanatory. Today, drug development is a complex process which has, I would say, a desperate need of integration of diverse research areas, including engineering, biology, chemistry, and pharmacology. With respect to such a need of integration of different pieces of knowledge, the career and the expertise of Paolo is, in my opinion, paradigmatic. Quite a long time ago, Paolo was a student in this department. In year 1992, received the Master of Science degree in Electronics Engineering. In 1996, he completed the PhD program in bioengineering working in Claudio Cobelli's lab. In 1996, he joined the Department of Bioengineering of the University of Washington, Seattle, where in 2001 he became associate professor. In Seattle, Paolo directed several research projects funded by NIH concerning the development of mathematical modeling methodologies for biomedical research. Afterwards, Paolo moved to industry. From 2008 to 2015, he was a research fellow with Pfizer in San Diego, California. Since 2015, Paolo has been senior director of clinical pharmacology and pharmacometrics at MedImmune, a subsidiary of AstraZeneca located in Cambridge, UK. In the meanwhile, Paolo received a Master in Business Administration from the University of Southern California. He published to date 130 peer-reviewed articles. He served on a number of institutional committees in the US, and he's on the editorial board of several major journals of the pharmacology research area. Paolo is a fellow of several scientific societies, and he is also fellow of the International Business Honor Society known as Beta Gamma Sigma. I believe that the audience today is quite heterogeneous, but I'm quite sure that all of you will be provided there with very interesting research insights. However, the proof of the pudding is in the eating. So let's give the word to the speaker, and please join me in welcoming Paolo.
Thank you. Yes, I think it is, yes. So good morning, Giovanni, Sandro, thank you very much. It's really great to be here, and I want to welcome the students, the faculty and staff, and all those of you that they were able to join us this morning. As Giovanni mentioned, this is going to be an overview of my personal path in joining engineering, biology, pharmacology in the course of my academic and industrial career. I hope that this will be useful to some of you to learn more about this field, and maybe it may inspire some of you to actually contribute to what I think is a very important problem. So Giovanni has already mentioned something about me. I spent some time here, of course, in Padova. It's great to be back. And then I joined bioengineering at the University of Washington. You can see the cherry trees over there. Spent some time in San Diego with Pfizer Global Research. And then I left there to join MedImmune a couple of years ago with their clinical pharmacology, pharmacometrics, and the MPK department. You probably don't know much about MedImmune, so I'll give you just a very short overview. It's a global biologic company. It's the biologic arm of AstraZeneca, and it has 3 sites that are geographically distributed, one in California, one in Maryland, and one in the UK where I am. What does it mean to be the global biologics arm of a global company? We work in the development of large molecules, that is biologics. That's sort of the top left, is where we are. There are 2 biotech units with us. We have the IMED, the small molecule area of AstraZeneca. We together feed into the business, which is called global medicine development, in the end, contributing to the pipeline of the global company. Probably not all of you know what the difference is between small and large molecules, but during the course of this morning, I will clarify what that means and what these areas of development of new drugs are. I would like to make this a bit interactive, so if you have questions or ideas or things you want to ask, please do that even during the presentation. It's really no problem. So like I said, it's really great to be back in Padova, looking at the speaker roster. I'm a little humbled, in fact, even more humbled because I'm speaking here in a lecture room which is named in the memory of Professor Lepsky, who I met in these hallways many years ago, and I really was always admiring his ability to teach and the breadth of his interest. So it's really great to be here, and especially in this lecture room. So let's start with bioengineering. Bioengineering is a very diverse field. I mean, we have, if you look at bioengineering around the globe, it's been able to develop technology of value to humanity in many different areas, from devices to computation to medical and clinical, computer science and imaging, just to name a few. To do so, bioengineering, as you all know, borrows on tools that come from fields that are very diverse: physics, chemistry, medicine, biology, chemical engineering, mechanical, electrical, which is where I come from. So my focus in this very diverse area is in how bioengineering, computational modeling, and simulation has contributed to drug development. In particular, the path from discovery to clinical science that leads molecules to become medicines, right? So it turns out that many contributions in modeling and simulation in modern drug development are inspired by bioengineering principles. And so that's something that I want to convey today, just so that people know, that there is room for engineering principles in how we develop new drugs. So the marriage between biology and engineering is really not new. We have seen, many advances in computer modeling of complex systems. Advances in whole heart modeling, electromechanical, physiological models. The reason why these advances are even possible is the improved communication between engineers and biologists in the ability to find a common language, a language to communicate. Where do the languages of engineering and biology actually meet. Turns out that biology usually summarizes their results with the help of diagrams, such as this one. Diagrams that are often not suited for quantitative analysis, for analysis that is rigorous and quantitative. The language of biology tends to be vague and sometimes avoid clear predictions. The language of engineers is very clear. It's built on standards. It's built on terms that are understood unambiguously by everyone that is involved in the discussion. Engineering language is suitable to modeling and simulation. So it's common experience that when the number of components in a complex system exceeds a certain threshold, I think it's 4 to 7, it becomes impossible to follow them in your mind. But the existence of formal languages in engineering, actually facilitates the design and the understanding of complex systems, be they aircraft or computer programs. So why is this important for drug development? Why is it important that we become more rigorous, more quantitative, and more able to ask precise questions of the systems they want to analyze? Because it's an expensive and a risky process, and any opportunity for improvement is very relevant. The cost of development is around £1 billion. I can go from pounds to dollars to euros, so hopefully you don't mind. So it's not unusual, in the late stage of development for 5,000 to 6,000 patients to receive the medicine. Each patient has to be monitored. It's a time-consuming and expensive process that can last a very long time, in excess of a decade. I don't know if you're familiar with the concept of pipeline, but when we develop new medicines, the molecules start at one end of the pipeline, and then they end at the other end, from discovery of the target and evaluation to launch of the molecule. The company pipeline can be a very brutal and harsh environment. You start with 5,000 candidates. Of these, fewer than one will progress to the pharmacy. Like I said, extremely risky and extremely expensive. So 5,000 to 1 can progress, to clinical testing. 5,000 to 5 will progress to that stage. So, like I said, opportunities for improvement are needed, right? So I'm going to tell you a little bit about how these happened in the last, I would say, 10 to 20 years. It's a process that I've been associated with starting in the early 90s. It's been a really good ride, to be honest, so far. So because this is a bit unusual for this audience, I will start with some of the principles. So I'll talk about PK and PD, and then I'll talk about some of the newer advances like pharmacometrics and systems pharmacology, which are sort of the new frontier where we want to push quantitative sciences in drug development. So what are some of these principles that scientists use to advance new molecules? I will start with PK and PD, pharmacokinetics and pharmacodynamics. It's really these principles hold the key to how we try to understand how drugs work in living biological systems. So briefly, PK is what the body does to the drug. So when you take a pill or take an injection, that drug molecule will be transformed in some way in your organism, right? The time course of concentration from dosing to clearance is going to be shaped by what you are, shaped by the bodies, by the circulation, by metabolism, by your organs, and so on. So the way that we choose to understand this is by analyzing the time course of drug disposition from dosing to final clearance, to last clearance. On the other hand, the new frontier, really the thing that brings biology together with the drug development, is pharmacodynamics, which is what a drug does to the body. So here, we try to understand how increasing drug concentrations lead to a higher effect. Normally, this profile will plateau, meaning that when concentration gets to a certain level, effect will saturate in some way, right? So these are the basic principles. When you have a new molecule, the idea is what happens to it, right? And what does it do, right, in the body? These are the basic principles. Drug action requires a drug molecule to bind to its target, right? The target can be a protein. In this case, it's HIV protease that I'm showing here. And the drug, this is nelfinavir, it's a protease inhibitor, binds to certain parts of the protease so that to block its action. There are many mechanisms of action. They're all different, but they all require that a drug finds the target and then binds to it, right? So the process of binding is dictated by the affinity of the drug for the target, and then where the target is in the body. As you can imagine, this changes depending on therapeutic areas, depending on where the target is, what the process is, and so on. But the idea is that the drug has to find the target, then bind to it, and do something. That's the basis of pharmacodynamics. Okay, these are basic principles that I think are needed to sort of talk about this topic in some detail. Like I said, it may seem simple, find the target, develop the molecule, find the optimal binding. It's not as simple as lock and key, of course. Discovery and development, like I mentioned, remains a very risky process. Why do some molecules fail? Why does this human endeavor of developing new medicines is so risky? There's so many reasons. Some of them have to do with cost and complexity of the disease target. Some of them have to do with the molecule itself. The molecule doesn't get where it's supposed to go, and so it won't work. It's not sufficiently selective for the target, has side effect. Competition is too far ahead. It's impractical to make, and we don't own the right intellectual property. So these are all reasons why this process can fail, and this is why something like predictive computer modeling and simulation can actually make a difference. Which brings me to the current state of how modeling and simulation, computer modeling, is applied in drug development. And this is a bit of a focus on regulatory milestones, from pre-IND, investigation of new drug application, all the way to biologic license application or new drug application, and then marketing and post-marketing surveillance. All these milestones have very important contributions from the modeling and simulation, which impacts now many areas of discovery and development, both in a molecule-specific manner that have to do with the particular molecule effect as it progresses through the clinical pipeline, and in a platform modeling scenario where we try to learn from several molecules at the same time. So the idea of using computer modeling has to do with the specific molecule that I'm studying, but also we try to learn across different mechanisms of action so that we can learn more and more about the underlying pathophysiology. And so I'll talk more about what we call now systems pharmacology, which is a systems view of drug action that borrows from biological systems and several drug molecules at the same time. Now, one thing that also is maybe important to bring up now is that the pipeline going from left to right, it's a bit misleading. In fact, this is not a one-way street by any means. We try to learn from late stages. We inform our projects in the early stages and so on, so that it's more of a continuum and a cycle as opposed to strictly a one-way street where we just execute a series of milestones. So, in the end, as shown in this example here, which concerns angiogenesis, the philosophy is to build bridges from exposure, PK, to response, PD, at the same time understanding all the individual steps that shape how the molecule goes from administration to effect in a subject population. Right? That's basically the goal that we have here. Now, it stands to reason now, and we'll talk more about it in the next few minutes, that drug dose, the actual number of molecules injected, is not by itself the primary predictor of effect. That may seem a little strange, but so many factors intervene between dose and effect, the PK, the PD, and so on, that there is substantial variability in how individuals react to drugs. So we may all be getting here the same dose of drug, but we may have different effects, different exposure. And the reason is that any one of these steps that I described, where the dose is shaped by the PK to concentration, and then PD, and then effect, every one of these steps is different, between each one of us. And so part of the goal and the problem is to understand exactly why we are different, what makes us different with regard to a given drug molecule, and how can we understand that difference in a way that helps us dose these agents optimally. So I'm going to start with the PK. I'll tell you a little bit more about the recent advances in pharmacokinetic understanding, just so that you can see with your eyes how engineering can help. Engineering principles have helped already in characterizing the fate of drug molecules. As I mentioned, PK is understood with reference to time. So the increase and decrease of drug concentration in the various locations of the body helps us become informed on ADME, absorption, disposition, metabolism, excretion of new drugs. This is done now with mathematical models. The models on the left can be relatively simple, complemental models. Many of you in the audience are familiar with that. The models on the right are a little bit more sophisticated. They aim to mimic the actual interconnection of individual organs as they're based in the circulation, right? These are, we call them physiologically based pharmacokinetic models, PBPK. You specify the organs, you specify their interconnections, you know the flows, and then you just run the simulation with your molecule. And these models are now ubiquitous. They are very, very common, especially for small molecules. The building blocks, use properties of the organism, properties of the drug, and properties of the study protocol. But now today, all of this is now part of one unique simulation environment. And now everything, of course, is made possible in this case by the computational platforms that are provided by software engineering and informatics. But this is already happening, of course. It's fairly routine for small molecules. In fact, one of the most common applications of this technology is to help us predict what the fate of the molecule would be in humans when we have only preclinical or animal data. So the idea is, again, it's an iterative process where you start building your simulation on a preclinical system, and then you predict more in humans and then in a population of humans, all the while learning from each data set in a process that we can call learn, confirm, and refine. So learn from the early data sets, confirm in the simulation, refine when there is a mismatch. So in the end, what you have is an integrated simulation environment that speaks to the properties of your specific drug molecule. Okay, that's I think the state of the art when it comes to PK. But let's not forget why we're doing this. Pharmacokinetics is not important per se. It's important insofar, it helps us understand how drug dose relates to the in vivo pharmacology. So the goal, in this case, is to find the optimal dose that leads to the optimal concentration that gives us the best fit with the known therapeutic window. That is, we want high efficacy and low toxicity, right? So sometimes these curves are very wide apart. Sometimes they're very close, in which case it may be bad for molecular progression. But the idea is to understand how the dose relates to the ultimate in vivo pharmacology. And through that, an understanding of pharmacodynamics, PD, is now required. So there's a lot of examples that I could make how PD is influenced by computational sciences. I chose one that I'm familiar with, which is translational oncology. So when we look, for example, at targeted anti-cancer compounds, the usual, I would say, paradigm to understand translation of these agents from animal to human is to model the time course of the drug, the time course of target inhibition. This could be the epidermal growth factor receptor or some other oncogenic target, and how the inhibition of the target in vivo relates to inhibition of growth of tumor lesions in a preclinical model. So the idea, again, is to go from dose to PK, how much drug is there in the tumor and in blood, how that drives the shutdown of the oncogenic target, and how the shutdown of the target then leads to inhibition of tumor growth, right? So again, these are temporal processes. We can understand them very well in preclinical models. Now the idea is, how do we use these preclinical rodent results to lead to clinical prediction? And so again, the idea is to map concentration and effect to the human system. Some of us have worked in how to best design translatable preclinical studies where the attempt is really to make the preclinical system, the animal system, as close as possible to the human situation. An easy way to do that is to make sure that at least the concentrations that the animal is exposed to are the same that the human will see. In doing that, in trying to match these exposures from animal to human, and in improving translation, we can be sure that the effect that we see in the non-clinical system is maximally relevant to the human situation, because we're not going to expose animals to doses or concentrations that are too high with respect to what a human can tolerate. So this can lead to an improved understanding of exposure response and how this will ultimately help in human dose prediction. So translational oncology in industry makes intensive use of these kinds of approach, where scaling occurs at the level of dose, exposure, response, and then effect. And it's very useful, especially for first in human, but it can be used depending on data also later in the process. Now, I want to go back to why the centrality of dose. I did tell you that dose is not the whole story, but of course, posology, the principles that underlie optimal dose determination, is an important task for clinical pharmacologists in industry these days. It will not be a surprise for this audience that the poison is in the dose. You probably have heard this many times. Sorry, it's me. Water, coffee, and alcohol are all poisonous when given in certain amounts. So since, like I said, the mid-90s, the industry has been using several approaches, including system modeling, statistics, and so on, to learn as much as possible from routine clinical data to optimize dose. Let me tell you a bit about PK in a population of subjects. So if we were all the same, and we were all getting the same drug dose, the drug concentration will be the same for everybody. But we're not all the same. So when we get the same drug dose, we're going to have different time courses. Right? We're all going to be different. So the exposure of the drug is going to change depending on who we are, even if the dose is the same. Then, of course, to complicate the matter further, every time we measure concentration, it's done at discrete events. And just to be absolutely clear, we don't have those lines when we try to learn about the molecule. We only have these sparse points. So the idea is how can we learn about what the underlying structure and variability was from this kind of sparse measurements that are taken very far apart in time and in a relatively small number of subjects. So we do that using a set of tools that we call population PK, or nonlinear mixed effects. Again, this approach brings together contributions from engineering, from computer science, informatics, and statistics. So the idea is that the computational model of drug disposition is not one for everybody, like the blue line, but it's different for everybody in a way that is dictated by important covariables, which could be demographics or disease state variables, and things that predict the disposition of the drug in vivo. So in how to tease out the influence of factors such as biometrics and demographics, we try to understand what makes each of us different and how can we adjust doses in a population when the drug is finally prescribed. So these covariables impact drug disposition. You can have, here just for illustration, I have how you would pursue understanding the effect of sex, creatinine clearance, or renal function, weight, and age on drug disposition. As you can see, there are some relationships that we can build into our models so that patients that live in this continuum, you can predict what kind of effect that will have on drug clearance or disposition of the drug, when it's dosed. If, for example, you have a trend with body weight, you can explore dose adjustment based on weight and so on. That's done for some molecules that are used these days. In the end, you have a full statistical picture of how drug disposition changes across population of subjects. Depending on who you are, you live in different parts of this curve. And all this comes from routine clinical measurements of exposure that are gathered in clinical trials, so that we can use these models then to predict typical drug disposition in a population of patients. So it's not what you look at matters, it's being said, it's what you see. So extracting patterns from these observations, is really important because it helps us understand about the underlying mechanism. And the objective, of course, to identify what the best dosing is. If you want to fit our concentration in a given therapeutic window, not too low, not too high, then these principles help us understand the frequency, the extent of dose, how important it is, what happens when a subject misses doses, which happens from time to time, and so on. So these are all helpful. These principles are all helpful, and they borrow intensively from engineering principles. So I introduce now a new term. We talked about PK and PD in some detail. We talk about population PK and how that borrows principles from statistics and engineering. The science of quantitative pharmacology has now been known as, is recently been known as pharmacometrics, which is basically the science of applying mathematical and statistical methods, such as those that we have described, to understand and predict PK/PD and its uncertainty. So these techniques can be used for, some of you asked, I was with the students yesterday, some of them asked, do you use some of these models for decision making? And I would say, absolutely. This is done at many different levels, both internally and externally. I have a table here, which is a little bit old, but shows the various regulatory decisions that are impacted by some of these models in the late stage: approval, labeling, trial design, and policy. And the reference here has an internal FDA survey on 42 new drug applications, where 26 out of 42 had a pharmacometric component, labeling-related issues in 37 NDAs, and pharmacometric analysis were pivotal in more than half of these 42 NDAs. So these analyses are valuable to gain insight on development, and they're used by many, many stakeholders to make decisions across the process. So I talked about the label a little bit. How do these analyses influence the label? I have here a biologic label. This is what you call the packaging insert, maybe. And it describes, as you can see, it describes the pharmacokinetics, the absorption, distribution, metabolism, and elimination. And it does so with numbers. How much is clearance? How much is volume of distribution? How much is bioavailability, right? These are all key parameters of disposition. And it's critical to note that pharmacometric analysis can actually contribute to how these numbers are calculated. In the end, helping everybody, patient, but others as well, to be more informed about how the drug should be prescribed. We talked about biologics, so I'm going back to that, small molecules to biologics, what is he talking about, right? So there's actually two main kinds of molecules that we operate with these days in modern drug development. I'm going to switch gears a little bit and talk about technology again and why modeling is important for biologic molecules as well. So for those of you that don't know about biologics, molecules that are used as active substances can be divided roughly in two classes. One is the one that classical drug development works with, small chemically manufactured active substance molecules. One example is here is aspirin, acetylsalicylic acid, which has a molecular weight of about 180 daltons. These are small, they can be processed into ingestible capsules that dissolve in the GI, and they can reach any destination in the body. On the other hand, biologics are large protein molecules, 25,000 atoms versus 21, 150,000 daltons versus 180. The same complexity that I think compares a bicycle with a jet engine, with a jet plane. So these are essentially copied or optimized versions of human proteins. There are various characteristics that distinguish them from small molecules. You're most familiar with small molecules because you can take them by mouth. Biologics have to be injected. They target extracellular targets versus intracellular. They stay in the body a very long time. Small molecules don't stay that long. They're very sensitive to heat. Small molecules are very stable. They are degraded by protein catabolism versus metabolism like small molecules. And of course, because they are so big, they can cause immune reactions in the body, so we call that immunogenicity. That's another differentiator between small and large molecules. Biologics are created by living cells, not by chemical reactions. We are used to think of drugs as chemicals. Biologics are not chemicals. They are actually derived from living material, often mammalian cells. It's a very complicated manufacturing process where, I should say, a lot of chemical engineering and biological engineering can actually play a role. the manufacturing of biologics is a very complex science that borrows, again, from many different disciplines. It's not my area, but it's certainly something that you should be aware of, as biomedical engineers, some of you. So the downside of the complexity is that because this is derived from living material, small differences in the production process can alter the finished product. And so, that being said, there is a big payoff for developing biologics because they're very versatile. They can be engineered to last longer in the body. You can combine them with different molecules. You can have biologics as a vehicle to carry small molecules in other locations in the body to target tumors precisely, like with antibody-drug conjugates. So this, for example, is one area where small and large molecules actually come together and can be both used to improve the therapeutic outcome. The other thing about biologics is that they are very specific to their target. They have very high specificity. They hit the target with very high affinity in the picomolar range sometimes. And they're different from small molecules because these can be very non-specific in terms of how they hit their actual target. Sorry about this. Like I said, I'm going to mention briefly, these have unique manufacturing device considerations. Pills versus injections, stable at room temperature, cold chain delivery, simple to make, very complex to make. So these are some of the things that I mentioned already, but it's really important that I would encourage you to learn more about how biopharmaceuticals are made, because there are areas of where principles that come from engineering can actually make a difference. It's an important opportunity, I think, for biomedical expertise. So there's more to biologics than size. What makes these interesting from an R&D point of view is not just the fact that they're bigger and they're more specific, but for biologic molecules, their pharmacokinetics, their disposition is inextricably linked with the biology of the system. That's what makes it very interesting, because you can't really understand disposition of biologics without understanding the underlying properties of the biological system. This is in addition to the other things that we already mentioned. Here's a biologic disposition model. You not only have the mere distribution, but you have additional routes of elimination that have to do with target binding and formation of complex with immune molecules. So when developing a new biologic molecule, one is to be aware that its disposition is going to be very complex and very different from what you would do with a simple chemical. And the degradation pathway, these are very delicate molecules. The degradation pathway that it can be subject to can be very difficult to measure and to control. That's why when we select some biologic molecules, we have to be very aware of how this will influence their fate in the body. Now, I talked about the aspect of mechanistic models. So why do we care about having mechanistic realism in our simulations? Because that will make our predictions more accurate. As I mentioned earlier, the field is in need of new approaches. We need more specific quantitative approaches that can make our job a little easier. So different from Ptolemaic epicycles, we want something that is real, that is actually mechanistically realistic. So again, the last area that I will talk about in terms of modern drug development and how it has benefited from computer simulation is the idea of systems pharmacology. So you all have heard about systems biology. I think many of you have not heard about systems pharmacology. But the idea of systems pharmacology is to integrate the understanding of the biological system that comes from systems biology with what drugs can do to the system. So it's the integration of pharmacological modulation and underlying biology. So systems pharmacology, I think, is the latest chapter in our quest to build quantitative sciences across the drug development continuum. So, as I mentioned, it's the interface between pharmacometrics, which I described, and systems biology, which is instantiated by making our models more realistic when it comes to the underlying biology. So, again, some of us have been part of this journey from the very beginning. In 2011, the NIH has released a white paper that talks about these approaches was the product of 2 workshops that were done in Bethesda, Maryland. And after the release of that paper, the interest in the field has really grown significantly. So this is just publications, but I can tell you that even internally to companies such as mine, this field is looked at with much interest. And it's considered to be, so to speak, the next frontier to help us guide our experiments and learn about the system a little better. What makes systems pharmacology different from systems biology? You all have heard about the fact that we look at that we can develop multi-scale models that look at different levels of biological organization. I started with the heart, remember, there are other examples. These models look at the biology of the normal state. They don't necessarily talk about pharmacological intervention. And this department actually is a long and distinguished history in modeling the glucose-insulin system and the flows that shape how these 2 molecules interact. Systems pharmacology takes a different approach. It looks at the system as the target of various modulations that come from drugs. So by looking at different drugs and how they modulate the system all together, the system pharmacology approach takes an integrated view of how the system works and how it can be changed. And so by using all these different probes and learning about them simultaneously, it's a tangential approach to the traditional systems biology to learn more about the system and how it changes over time. Not by looking at the underlying fluxes, but by looking at how drugs change those fluxes. So one example here is hyperglycemia, how different targets across the spectrum actually can modulate that. But there are examples in cancer as well. Cancer is a very complex system. And so by looking at how different molecules such as EGFR inhibitors, PARP inhibitors, inhibitors of MET, telomerase inhibitors, and so on, how do those change the fate of individual cancer cells together? We have a systems pharmacology view of cancer cells, and where all these interventions can be deployed at the same time to understand the interplay of their growth mechanisms. All this, I'm showing you pretty pictures, but all this can be used and deployed with a computational system as well. And now to add to the complexity, some of you may have heard that many of us are looking at immunological interventions to improve the outcome of cancer. Immune modulators are the last frontier in oncology. And so one could take a systems view of immune modulators as well, where you look at cancer immunity cycle and how some pharmacological interventions can impact both the underlying cancer system and the drugs that touch the system, and the immune cells that eventually can remove cancer cells if they are instructed properly to do so. So something that I mentioned already. However much we would like to simplify their mechanism to facilitate our understandings, biological systems do not operate according to simple rules. It's really about calculus, it's not about arithmetic. And it doesn't matter how sophisticated is your experiment, you will not tease out those complex rules. So mathematical models are useful to interpret those signals, and the corollary is that we can use these to progress our molecules, to make decisions, to help us understand what the molecules actually do in the quest for optimizing the delivery of drug molecules. Now, these are some of the things that I mentioned earlier: PK/PD, pharmacometrics, systems-based approaches, and we think they all have a place in how we progress from target selection to molecule selection, in vitro profiling, preclinical experiments, clinical testing, and then finally medicine. So all these are methods. They have a place in this continuum. This department has a long history of developing methods such as those, and it's been very interesting to see how these approaches have actually been used in practice in what I've been doing over the last 10, 11 years or so. So I want to leave you with some examples. This is actually happening. I have been involved, for example, in an interesting systems pharmacology approach to tumor immunotherapy in collaboration with Johns Hopkins University, which was meant to capture the interaction between immune cells and cancer cells in the context of what we call the immune checkpoint inhibition approach. So this is what is called an agent-based model. It's basically a self-learning or evolving system that models the cells as individual agents that interact according to specific rules, in the end giving rise to complex emergent behavior. And the beauty of the 3D simulation is that we can take slices at the computer and then compare these slices with histology to help us understand where these cells go on the computer and where they go in the actual system, so that we're being very pleased by how this approach has helped us understand some of the dynamics of tumor immune cell interaction. When I was at Pfizer, we worked at a computational model of how protein drugs can engender an immune reaction. So again, this was an interesting approach with a multi-scale model at the subcellular, cellular, and whole body level for drug disposition and immune system triggering. That we were able to validate that against preclinical data and some early validation against clinical data. And others have used this to deploy with their own molecules, which actually it's an important part of how we communicate these days. Many of these models are actually made available so that you can learn from what others have done. They are published, they're made into repositories that you can query. And so the access is probably larger than it ever was, because some of this code can actually be downloaded and used on anybody's computer. Coagulation cascade was another problem where I was involved in. And this is, again, a multi-scale model that describes how the coagulation cascade works and how several molecules actually impact it, right? So, and it was useful to help us identify some markers or measurements to monitor, to predict the response to different therapies. So these three examples, give you an idea of how wide the range of approaches, of applications that you can have for these approaches. And the underlying technology, is the same. It's modeling, differential equations, engineering principles. But the application, is really very diverse, from metabolism to cancer to coagulation to immunogenicity. So it's been a really good ride to see how these methods and approaches can really make a difference in decision making. So I want to bring it all together by telling you what the view is now in terms of how these approaches would integrate. I talked about PK/PD, a relatively empirical view of drug action and disposition. I talked about systems pharmacology. So these have been used for a very long time, and they're used to increase your confidence in the molecule, right? Systems approaches that bring together the biology and the pharmacology help us understand the target, so that when they come together, we increase our confidence in the proof of concept the drug is actually doing what it's supposed to do. So focused on the molecule, focused on the biology and the target, they have to come together to help us make better decisions, as we progress new molecules towards the clinic. Here's a little story, that I want to tell you, about how we can be better at this. This is a study the Boston Consulting did 842 molecules over 2002 to 2011. And they looked at what factors were correlated with the success of these molecules, right? So what made these molecules successful? Was company R&D spend, was where the company was, market size, therapeutic area, target family, none of these. Even surprisingly, molecular properties. What actually was predictive for success were indicators of scientific acumen. So publications, citation per publication, patents per R&D dollar spent, right? Measures that even in academia are considered objective measures of scientific acumen, right? So surprisingly, or maybe not, being a good scientist correlates with success in drug development. The data show that. And I'm reminded of something that I heard many times ago from Lou Scheiner, who was one of the pioneers in drug development approaches. He released an interview where he said that when he talked about productivity, he said that to be productive, scientists need to keep their eye on the ball, on the problem, which is understanding the subject matter better or teaching students better. Then everything else falls out. They become successful as a researcher or successful as a teacher and get the rewards. But they should not keep the rewards in mind as the reason for it. So we shouldn't really focus on the ultimate reward. We should focus on the science. And so, of course, change happens in science like in everything else, and change is not necessarily a good place to be. Change is probably going to accelerate even further. When it happens, it maybe seems scary, a little bit unsettling, but it's the nature of the world that we operate in, right? We need to be able to change, to adapt quickly. And of course, us and many like us are looking at new areas of interest where where we think the future is going to, be impacted by. We are looking at things like in vivo expressed biologics, where we are trying to have our own bodies actually make the drug molecules as opposed to, biotechnology manufacturing. Precision medicine, which is focused on how to develop best medicines for certain populations of patients. And of course, digital health, which is anything that improves the patient experience, either in trials or in connecting devices and predictive analytics and things like that. These are some of the areas that I think the industry will focus on in the future, going forward, which will again contribute to these important advances that I talked about. So I hope you enjoyed this little brief overview of some modern application of engineering principles to pharmaceutical R&D. From my, this is largely a personal overview. It's my own point of view. Of course, it benefited from the contributions of countless friends and colleagues. Some of them are actually here in this room, and I was very fortunate to work with all of you over the years. So, maybe the final message, and my experience has been that to have a role in drug discovery, you need to be, so to speak, a jack of many trades and a master of at least one. There's new models of drug discovery that are being developed. It's a time of exciting, new advances, non-traditional targets, better ways to look at information, bioinformatics approaches to how to process new information. And to me, it's very exciting. It's a very multidisciplinary endeavor. Pharmacology and biology, but of course, engineering, computer science, bioinformatics are required. We're not, project teams are the norm in the pharmaceutical industry, and it's been very exciting, to see these form and communicate, and it's exciting time to be developing new therapeutics, and I hope I could communicate some of the excitement, to you today. So I just want to thank you very much, for being here so long. Thank you.
Thank you, Paolo, for this brilliant lecture.
Thank you.
I believe that the necessity of bridges among disciplines was very clear in your presentation. Yeah. So I expect many questions from the audience. So
I'll be around in case you wanna. Maybe I wonder if we can turn the air conditioning on back on again.
Who would like to be the first one?
Thank you very much for your very, very interesting presentation. I'm a chemical engineer by background, and I have a limited experience in working with pharmacokinetics and pharmacodynamic modeling. But one major occurrence that I came across with, and you have mentioned it quite clearly, is the diversity of responses of individuals to a given molecule. And I wanted to know from your point of view how this variability can be addressed at the modeling side. You have spoken about models for populations. Yeah. My experience is that it may be very tricky using models for populations because single individuals may respond in a totally different way. So how far are we from developing individual models and from developing, let's say, clinical tests that can be personalized in order to identify the models for single individuals rather than for populations?
No, thank you. It's a very important problem. So there's two facets to your question. One is how do we understand what shapes variability, And then what do we do about it, right? So how important it is that we address variability. So as far as the first part, there's a growing corpus of approaches where we try and dissect the variability out. So you can think of this as drug disposition being a big cloud, right? And then you add in, say, body weight, you add in maybe age or sex, and all these variables sort of shrink the cloud, right? Because now variability becomes a little less because you are using these variables to explain what previously was random, right? And now some of it is deterministic because the drug is cleared maybe more quickly by subjects that have higher body weight, right? And so that becomes, and so the variability shrinks. So the statistical techniques that I talked about can be used, to address that problem. And they're usually applied to differential, so they can apply to any model that is linear or differential equation, doesn't really matter. But they can be used to dissect this variability. Now, when does this variability translate in the need for dose adjustment, maybe for specific subjects? So we want the effect to be substantial, right? So I think that every time that, and there are some antibiotics that have individualized dose adjustment of some kind, every time that this makes a difference for patient outcome or safety, we have to be sure that that is incorporated. But we want that to be substantial, like a big difference, right? That would have really an impact. But there are statistical techniques that can help dissect the variability. And the basic idea is that what was random now is at least partly deterministic, right?
Professor Ruggeri.
Thanks, Paolo, for the very interesting presentation. I have a question which is more a curiosity than a technical question. When you move from very simple model like PK, PD, 1-2 compartments, they're very simple. You can develop them in a very easy way.
Yeah.
When you move to system pharmacology, then models become very large, very complex, and I guess very expensive and time-consuming to develop. On the other side, many pharmaceutical companies are working on the same type of disease or the same application. Is there any kind of project where you can develop models very complex and can be shared among different companies?
Yeah.
So just to avoid or to at least cut the time, which is important in drug development, and the cost to develop these models.
Yeah. No, it's a good point. So my experience in industry is that industry can be very open. In terms of pre-competitive collaboration, is certainly something that we have done, and we totally would encourage it, especially when it comes to these complex models. The immunogenicity model that I mentioned, when it was published, the code was released, and I'm aware of at least another company that has used it internally for their own validation purposes, and now another outfit is taking the model and developing something else with it, which I think is great. I mean, after all, most of the time, pharmaceutical companies are not interested per se in the development of software or code. It's not something that we would, unless it provides some sort of a competitive advantage, which is very rare. So I would say that pre-competitive collaborations are actually exceedingly common. They may not take the shape of consortia, although those are out there. There are examples of that, especially for PBPK. There is a consortium that is been going on for a long time that is very active. But maybe sometimes even just exchanging the code or giving advice of how the code can be used or how the model can be extended, this happens quite frequently. So yes, there are quite a few opportunities to do that.
I am not in the field. I'm a system engineer, and so I'm allowed to ask a naive question.
No such thing.
No, the question is the following. Typically, in engineering, we are used to deal with quite simple processes, and that can be well described by simple models with a few parameters. And so you have also the possibility to have a lot of measurements and be able to validate and to have a good tuning of the model. While in pharmacology and biology, you have the need to have very complicated models, very few data. And so, in principle, for someone like me, it seems to be you want to solve an impossible problem. And the question is, how can you really find a way to make the model meaningful and validated when it is so complicated, and so you need to make simulations, and what you want is really to have a predictive model, and so quantitative models, but you have so few measurements, so few data, and so few reason to say that the model is good. Yeah. So what is the statistical methods that...
so it's a very good, I probably need another lecture to cover that. It's not naive at all. So the idea of model validation, so we do external validation. For example, it's not uncommon for one of the models to be built on a certain clinical trial and then be used to predict another trial that hasn't been part of model building. That's quite common. Predictive validation, predictive checks, we do those apart from the obvious goodness of fit. But I think you hit the nail on the head when you said that it has to be predictive. We like to use the word fit for purpose. So what is the model for is probably more important than what it does. So if it is meant, for example, to help us design a phase 3 trial on the basis of phase 2 data, we're going to use it for the purpose only. And we convince ourselves that it is appropriate for that purpose. Whether we want to expand it beyond that is not something that we normally do. So our realm of validation can be very limited, which is probably one reason why we can use these complex models and believe that they can be helpful. And in fact, they can be. But some of the traditional statistical techniques that we mentioned are used quite extensively. So we convince ourselves that the model is sufficiently robust for the question that we intend to ask. Does that help? I'm going to make, if there are no questions, I'm going to make one comment that is directed to maybe students and postdocs, that are in the audience. I know there are some of you. When I was at the University of Washington, it was very interesting because the postdocs in particular, were some of the population that was most difficult to engage, in the broader campus community, because they were so focused on their particular problem, their particular question or issue, or the laboratory experiment, and so on. And you would have Nobel Prizes, come to campus, and they would never have time to come. So thanks for coming today. I know how busy you are. So, but I guess what I want to say is that it's very important at this stage of your scientific career to learn as much as possible, not only about what happens within your area, but what happens outside in the broader scientific world. I had no idea when 20 years ago I sort of started dabbling in modeling and simulation for biological systems, they would turn out to be so influential in something as complicated as drug development. So it's something that I wasn't able to predict. But thankfully, I was involved in an environment and in an area that helped me sort of become attuned with those changes as they were happening. So try and reach out from your area as much as you can and be truly interdisciplinary, because a lot of how this works today is by being very interdisciplinary.
Any other comment? Professor Pedersen?
Thank you for a nice talk. I have a question that was related by your comment before on modeling choice. So in the multi-scale models, how do you work? Do you usually create a model for a certain level, say the subcellular, another model for the whole body level? And do you have these models speak together, or are they separate models for each problem?
No, it's a very good question. So when it comes to multi-scale model development, I think it's something that nobody knows how to do it right these days. I think we're all experimenting with different approaches with how to do that. I can speak to this one, which we're involved in. These 3 levels were assembled separately, but from day one, the interconnection between subcellular, cellular, and whole body was a topic of discussion. So you wouldn't actually, we would always try and develop the interconnections at the same time as the individual models, because we knew where we wanted to go, which was the whole body prediction of immunogenicity to therapeutic proteins. So that was our end game. So the level of detail and the attention to the subcellular and cellular level were dictated, by that purpose. So, and this is a differential equation model, which is what the audience is familiar with. This model here is an agent-based model, which is an entirely different approach, where every individual cell family or cell type is an agent that would interact with other agents. And so when you start the process of agent two-agent interaction, you never quite know what is going to happen because the rules are very simple. It's like a flock of birds. Don't get too close to the next bird. Look forward to the birdie from so on, and then they flock, right, in an orderly fashion. Something very similar happens here, which is a bit of a different approach. So, but the interconnections are crucial. In fact, they should be front and center when the individual levels are developed. That's my experience, at least.
So, if not, I would thank you all for coming, and really, I think the deepest gratitude of the department to Paolo for this great effort he made in coming here and delivering such an impressive lecture.
Thanks, everybody.
See you soon, I hope.
I hope so. It's been a long time, but maybe, who knows. I can do it more often. Thank you very much for being here.